Hello, everyone. Thank you very much for joining us today. My name is Filippo Petti. I'm the CEO of Celyad Oncology. I'd like to thank the organizers of the 2020 Cell and Gene meeting on the Mesa for allowing us to participate in this year's event. I will be providing you a little bit of background on Celyad Oncology. I will refer you to our SEC filings for additional background information on the company. So Celyad Oncology is a CAR-T focused company. We're based out of Belgium. Our mission and our vision is really to develop innovative cell therapies for the treatment of cancer. And our longer term focus is really to eliminate cancer and improve patient lives and the overall lives of individuals who are impacted by the disease. From a company overview perspective, we are led by a broad platform of discovery and development off the shelf as well as personalized CAR-T therapies. We are a leader in the NKG2D biology space, and really our lead clinical assets are NKG2D receptor CAR-Ts. We are fans of the NKG2D receptor. We think this is a differentiated approach to the treatment and to the development of CAR-T candidates, as well as the treatment of cancer. Given the fact that NKG2D, the receptor itself, targets 8 different stress ligands that are expressed on both hematological malignancies as well as solid tumors. And we think about the NKG2D receptor as really a pipeline in a program. We are advancing the field of allogeneic CAR-Ts with 2 proprietary technologies, what we call our TIM technology, as well as our shRNA platform. And this really leverages the work and our focus and strategy in using an all-in-one vector approach with regards to the design, the development, the manufacturing, and hopefully the eventual commercialization of our CAR-T candidates. And it really helps to simplify things. The all-in-one vector approach allows for the expression of both the CAR technologies such as TIM or shRNA, as well as additional add-on elements, all within a single vector. And we see efficiencies there in terms of how we think about the actual modality. We are underpinned by robust intellectual property, both for our allogeneic as well as our NKG2D CAR-T therapies. And we are supported by a GMP manufacturing facility that sits across the street from our headquarters in Mont-Saint-Guibert, Belgium, which is about 30 minutes south of Brussels. Turning to our pipeline, as I noted earlier, we are focused both on allogeneic, off-the-shelf, as well as autologous and personalized CAR-T therapies. Our lead program is the CYAD-101 program. This marries really our investigation of NKG2D, the receptor, and we're evaluating this in a metastatic colorectal cancer patient population. We are currently in a dose escalation study, and we are moving towards a dose expansion cohort of the AlloShrink trial, phase 1 trial, later this year. We're also evaluating additional next generation non-gene edited allogeneic CAR-T candidates. I will focus your attention to the CYAD-200 series, where we're led by the CYAD-211 program, which is a BCMA CAR-T for the treatment of relapsed refractory multiple myeloma. We recently received IND clearance for our phase 1 trial for CYAD-211. We plan to begin enrollment in the trial by the end of 2020. And the 200 series is special to us because it leverages our shRNA platform. And the 211 program is the first of what we believe will be many CAR-T candidates that will come from the program that will look at also next generation approaches where we can use shRNA technology to knock down and attenuate perhaps other proteins and genes of interest for these candidates. Our autologous franchise is focused on our CYAD01 and CYAD02. This is a first-in-class and next-in-class next generation NKG2D receptor-based CAR-Ts. Both candidates are now in phase 1 development for the treatment of relapsed refractory acute myeloid leukemia and myelodysplastic syndrome. Just quickly on NKG2D and the receptor, this is an activating receptor that is expressed typically in the natural killer cell. What we do at Celyad is we express this receptor in a T cell. And really the importance of that is because the NKG2D receptor itself is important with regards to protection around infectious diseases and cancer. This is really a novel approach to CAR T development because the receptor targets multiple stress ligands, as I touched upon before, the main bad actors MIC-A and MIC-B. What's nice about the NKG2D ligands, which the receptor targets, is that there's low expression in normal tissue, and it is seen and highly expressed in both hematological malignancies like AML and solid tumors like colorectal cancer. And there's other cells within the tumor microenvironment that also express these NKG2D ligands, these stress ligands. And those include regulatory T cells as well as myeloid-deprived suppressor cells as well. And we do see that these ligands are also on tumor neovascularization cells. Again, as I touched upon earlier, these form, the NKG2D receptor forms the basis for both our allogeneic 101 candidate as well as our 01 and O2 clinical candidates for AML and MDS. Turning to the CYAD 101, just to give you a little bit more flavor into what that is. So CYAD 101, it co-expresses, as I noted earlier, NKG2D as a receptor. It expresses our novel TIM technology, which stands for T cell inhibitory molecule. And we also co-express a selection marker to positively select the allogeneic cells that we create. This happens all in one vector. So again, the all-in-one vector approach to keep things simplified. We do have a single transduction of this vector, and it really leads to less manipulation of the cells and provides an opportunity to cut down costs and enrichment for those cells at the end process. The expression of TIM has resulted in our preclinical, and I'll take you through some of the clinical data, and really reducing the signaling of the TCR complex through a competitive inhibition model of competing with CD3 zeta, one of the components of the TCR complex. The AlloShrink phase 1 trial kicked off in 2018, which evaluated CYAD 101 following FOLFOX as our preconditioning chemotherapy for the treatment of metastatic microsatellite stable metastatic colorectal cancer. On this slide here, you'll see the initial data from the AlloShrink dose escalation trial. On the left-hand side, you'll see the typical regimens that are used for the treatment of metastatic colorectal cancer. These patients have typically seen some type of oxaliplatin-based therapy, FOLFOX, FOLFIRI, additional regimens that are used for the treatment of relapsed refractory metastatic colorectal cancer. Typical mutational status such as KRAS and BRAF is noted as well. In the center of the slide, you'll see a swimmer's plot. 15 patients in total. We dose escalated at 3 doses here for the trial at 100 million cells per infusion, 300 million cells per infusion, and 1 billion cells per infusion. And you'll see that we've had a few partial responders in the trial, as well as some durable stable diseases. With regard to key takeaways across the studies themselves, this was a first in human trial. We showed that we did not see any graft versus host disease within the first 15 patients that were treated in really first 34 infusions of the therapy, which is certainly provides a validation of the TIM technology and using a non-gene-edited approach to create allogeneic CAR T cells. We did not see any dose-limiting toxicities, as well as the tolerability looked good with regards to treatment-related adverse events. We did not see anything above grade 3 or grade 4. With regards to overall responses, I did note the 2 partial responders, a few stable diseases. In overall, 9 out of the 15 patients did show stable disease, 7 of those with 3 months or more, 2 of those with 6 months or more. So overall, there was a disease control rate of 73%. And we've reported a median progression-free survival of 3.9 months. Next steps for the program is to begin enrollment of the expansion phase of the AlloShrink trial, where we'll look to evaluate an additional 12 to 15 patients at the 1 billion dose level, and that is set to begin by year-end. Talking about taking next steps around our allogeneic programs, our CYAD 200 series. This is focused on our shRNA platform. We brought in the shRNA technology through an exclusive agreement in 2018 with Horizon Discoveries, and really leveraging their smart vector technology. Again, this takes advantage of a non-gene edited allogeneic approach and leverages the data we've generated so far on the TIM side and really our understanding of non-gene edited approaches. This is shown to have flexibility. We are able to knock down multiple proteins and genes, and I'll walk you through that a little bit. And we're excited about this because, again, focused on a simple approach and using all-in-one vector. We have seen in our preclinical models that the shRNA approach and knockdown is similar to a CRISPR-Cas9 knockout with regards to in vivo protection of GVHD, and we have seen some perhaps potential benefits in terms of persistence of the allogeneic T cells when using shRNA as compared to gene editing technologies from our preclinical data. Turning to the next slide, this is just a glimpse of some of the data that went into our IND application for CYAD-211. Again, this is a BCMA CAR T that uses a single shRNA hairpin to target CD3 zeta. And we've seen from the data below that we are able to control TCR activation using the shRNA technology, similar to CRISPR-Cas9, as I highlighted earlier. We do see graft versus host disease. We see protection against those as when compared to a control T cell using the CYAD-211 candidate. As well as on the anti-tumor activity on the right-hand side, you'll see that we are showing anti-tumor activity in the BCMA CAR T as compared to control and vehicle. So we're excited about the CYAD-211 program. This is really a concept to a clinic in 2 years. So I think is a testament in terms of the potential of the shRNA platform. And we plan to begin enrollment of a phase 1 trial evaluating the CYAD-211 program by year-end 2020. And just to quickly highlight some of the potential versatility of the shRNA platform, this is just a quick slide that shows multiple genes that we are able to knock down using an shRNA RNA interference platform. And you can see from the RNA levels and the protein levels that we can target multiple genes simultaneously, up to 4 in this experiment. And we think about that as potential next steps and next generation candidates for us, and thinking about a multiple shRNA knockdown approach where we can pull down and knock down elements such as beta-2M, kinases, CD3 zeta for allogeneic purposes, and perhaps proteins such as CD52. So versatility is certainly one of the benefits of this platform, and we'll look to continue to put resources and our efforts into evaluating the full potential of the shRNA approach as we move forward with our candidates. Quickly turning to our CYAD-01 and O2 program for AML and MDS. As I noted earlier, both the CYAD-01 and CYAD-02 are in phase 1 development. For the THINK trial for 01, we are in the expansion cohort, and we're looking to enroll an additional 10 patients there. Enrollment began earlier this year. And we plan to have an update for the THINK trial by the end of 2020. On the CYAD-02, this is our next generation candidate. We are looking at a dose escalation trial that we started in the beginning of this year. We have entered the 3rd dose level of the program, and we'll have an update from the dose escalation cycle 1 trial by year end. With regards to this next generation approach, which also uses some shRNA technology, specifically to target the MIC-A and MIC-B. And overall, I will say that the O2 program is shown in our in vivo experiments to have longer persistence, increased anti-tumor activity that has us excited about it in the context of treatment of AML and MDS. Just quickly moving to our upcoming milestones and financials. For the second half of 2020, we'll look to begin, as I noted earlier, the expansion cohort of the AlloShrink trial to evaluate CYAD-101 in the treatment of metastatic colorectal cancer. We'll begin to move forward with the 211 program and initiate our first in human dose escalation phase 1 trial by year end. And we'll have an update for both the CYAD-01 and O2 candidates for the treatment of relapsed refractory AML and MDS. And we plan to report data for both of those trials, the THINK and Cycle-1 trial respectively, by the end of the year. And as we turn to the first half of next year, we should have some preliminary data. We anticipate that for the 101 program in the expansion phase, as well as some initial data from the proof of concept side of the 211 study, at least helping us to better understand the potential of shRNA in becoming a novel allogeneic platform, and thinking about next steps for the 211 program towards the second half of the year. And lastly, just to quickly provide a quick financial snapshot for everyone, we ended June 30th of this year with roughly $30 million. We have publicly disclosed that will support the company's activities into third quarter 2021. And just to quickly reference, we are listed both on the NASDAQ as well as the Euronext Brussels and Paris exchanges under the ticker symbol CYAD. With that, I'd like to thank you for your time and look forward to speaking with folks in the future.